welcome to Professor SNAB, where we make A-level biology as simple as a cell. So today we're looking at the structure of bacteria and viruses, and we'll look at different diseases for each. So for example, for bacteria, we'll be looking at tuberculosis, and for virus, we'll be looking at HIV. So bacteria are prokaryotes, which makes them structurally different from eukaryotes, and they do not have a cytoplasm. The ribosomes found in bacteria are smaller, and they are described to be 70S rather than 80S, which is what you would see in eukaryotes. They do not have a nucleus, and instead they have a single circular bacterial chromosome that is freely suspended in the cytoplasm. This chromosome is not associated with protein synthesis, and the cell wall in bacteria, if present, is made of glycoprotein, and this glycoprotein is called peptidoglycan. It also contains cell membrane that folds, and this folded sections of the cell membrane is called mesosomes. They can also have a loop of DNA called plasmids. So these are just different features you are probably familiar with from GCSE and also topic 3. So if you do want to revise topic 3, it's already been posted. So do have a look at that. So going back to this, so sometimes they also have a capsule which protects the bacteria from drying out when attacked by cells of the immune system of the host organism. Sometimes certain types of bacteria also have flagella. This is a long tail-like structure that enables movement in prokaryotes. They also have thread-like structures called pili on the surface of bacteria, and this enables attachment to other cells or surfaces and also as a medium for gene transfer. So now we're looking at viruses. So they are non-cellular infectious particles, and they are smaller than prokaryotic cells. They have a nucleic acid core, and their genomes can either be DNA or RNA. And another thing is they can either be single-stranded or double-stranded, depending on the type of virus it is. They do not have plasma membrane, cytoplasm, or ribosomes. They do not have the typical features you would see in other types of cells. Some have an envelope that is formed from the membrane phospholipids of the cell they were made in, and some also contain proteins inside the capsid. They also contain attachment proteins which stick out of the envelope, and viruses depend on the protein building machinery of the cell they infect so that they can reproduce in numbers. So now we're looking at tuberculosis, which is our example of a disease caused by a type of bacteria. So this condition is caused by a type of bacteria called Mycobacterium tuberculosis. And before we go into further detail, we need to look at the definition of a disease. So a disease is illness or disorder in the body or the mind that leads to poor health. And they can either be infectious or non-infectious. Infectious is caused by pathogens and are transferred through vectors, as in they need a medium to spread between individuals. Pathogens can be bacteria, fungi, or viruses. However, it is to be noted that not all bacteria, fungi, or viruses are pathogenic. In fact, for example, some bacteria and fungi can be beneficial. Like, for example, the bacteria in your gut is beneficial to the body. So one example we'll be exploring in depth is, like I said previously, the Mycobacterium tuberculosis, which is the type of bacteria that causes tuberculosis. So tuberculosis is spread through droplets of liquid and can take place through actions such as sneezing or coughing. On entering the lungs, the TB bacteria is engulfed by phagocytes. And over time, the infected phagocytes will be encased in tubercles in the lungs. So this keeps the engulfed bacteria dormant. In this case, the bacteria can be active when conditions are met, such as the immune system being compromised by other infections such as HIV. The initial symptoms of tuberculosis include fever, fatigue, coughing, and inflammation of the lungs. And this can cause extensive damage to the lungs if left untreated, eventually leading to death due to respiratory failure. It can spread to other parts of the body and can cause organ failure in the affected organs as it progresses. So it is a deadly condition. So now we're looking at HIV. So HIV stands for Human Immunodeficiency Virus, and it is important in your exam questions you don't write HIV virus because that's the same as writing virus twice. It's a common error that you should be looking out for. So HIV contains RNA and is a retrovirus. So retrovirus is the kind of types of virus that can make DNA from RNA through a mechanism called reverse transcriptase. The vector for HIV is body fluids, and the various mediums are intercourse, blood donation, sharing needles, and it can be transferred from mother to child through the placenta, and sometimes even in blood mixing between the mother and the child during childbirth, and also through breast milk. So the virus infects the helper T cells, and these cells are the type of white blood cells that is responsible for activating antibody producing B cells. So from that, you can clearly see why it affects the immune system, because it's the B cells that produce the antibodies, which is what you need to fight different conditions, different pathogens in the body. The glycoproteins on the surface of the virus binds to the CD4 receptors on the surface of the T helper cells. The viral envelope fuses with the cell membrane of the cell, and the viral RNA now enters the cell. So reverse transcriptase is carried out using the cell's mechanism, resulting in the production of the corresponding DNA. This DNA is then converted from a single-stranded DNA to a double-stranded molecule and is inserted into the host DNA. So now what happens is the cell is going to produce multiple copies of this virus, because the DNA of the virus is replacing the actual DNA of the cell. And these enter the blood and are transported to infect other helper T cells, and the process repeats. The affected individual is now declared HIV positive, and definitely in the early stages, you will notice flu-like symptoms. So after this initial infection period, when the HIV replication is rapid, the replication rate begins to drop. And this period is called the latency period. In this stage, the individual will not show any symptoms. The number of helper T cells will drop, and this results in B cells no longer being activated and no antibodies being produced. And this reduces the body's ability to fight infections, eventually leading to AIDS, which stands for Acquired Immune Deficiency Syndrome. So I think, so when I kind of talk about the different stages that HIV goes through and the different stages that the that tuberculosis goes through, what I notice is that, like, you can clearly see that there's a difference in for example, in the case of HIV, there's a difference in the number of T helper cells, and it can quite easily be produced as a graph. So it is quite easy to sort of predict a possible exam question where you might be shown a graph that shows how the number of T helper cells in the body is changing over a certain period of time, and they might ask you to infer what might be the cause of the results in the graph. So then you can obviously infer that perhaps a sudden decrease in T helper cells could be the result of the individual being affected by HIV. So again, you sort of think about this information both for HIV and TB, kind of like how you can see it as a graph, or even like draw out a graph, like a simple graph showing how the number of different things that change in during the time period of the condition, so that you can sort of prepare yourself for those kind of questions. I don't think I've seen this question specifically before, but it's possible. So, after infection, the patient suffers mild flu-like symptoms, and after several months or years, the viral DNA replicated by the HIV particles become active. The virus particles eventually destroy and reduce the number of T helper cells present in the host, and this results in the patient no longer being able to produce antibodies and are unable to fight infections. So, for example, in this case, you could be shown a graph of how the number of antibodies change in someone's body, and they might ask you to infer possible reasons why this has happened. Again, this is just a hypothetical question to prepare for. And this results in the patient being more affected by minor health issues than typical. And such diseases are called opportunistic. And TB is also another example of such a condition. The condition will be classified as AIDS when the T helper cells count drop below the critical level and the patient is suffering from constant opportunistic infections. Again, if you see a graph with like the T helper cells dropping below the critical level, if it's shown on the graph, again, you can infer that the patient now has AIDS. So sort of try to use this video to look at different points and see how you can plot it as a graph to see how the number of antibodies, for example, changes. So these opportunistic diseases eventually leads to the death of the patient, and the time it takes for the condition to progress from HIV to AIDS and the survival chances is dependent on various factors such as the number of existing infections, the specific strain of the HIV, the age of the individual, and access and quality of healthcare. So that is all from Professor Snap today. Hope you found this video useful. I would recommend watching this video 2 or 3 times to fully understand both the diseases. And of course, if you have any questions, let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.